So terminal buttons need some things that can only be produced in the soma, and sometimes neurons are very, very long. In any event, to get material from the soma all the way down to each terminal button, it requires considerable, some sort of an energy-dependent process. And so that's where exoplasmic transport comes in. And this is how we get or shuttle materials down the microtubule system. It's, again, this active process that propels the various substances down the microtubules. There's one type of exoplasmic transport that goes from the soma down to the terminal buttons, and that is called anterograde exoplasmic transport. And it uses a protein molecule called kinesin, which kind of resembles a pair of legs and feet. And it walks down a microtubule tract to deliver its cargo to the destination, and it's very, very fast in terms of cellular processes. It can move material up to about 500 millimeters per day. And energy is provided in the form of ATP produced by the mitochondria. And here I think I should have a link to, this is a kinesin protein that's walking along a microtubule and carrying a synaptic vesicle. And that's essentially how it moves along. It kind of walks just like that. So it's not too much of an exaggeration. Oop. All right, stop. And this is an actual video of that process taking place in hippocampal neurons. And you can see it's really moving things along fairly quickly. So to get material back from the terminal buttons to the soma, that is usually going in the reverse direction, is not as fast. It's actually only about half as fast, and that uses a different protein molecule called dynein. So we have kinesin that resembles a pair of legs and feet, and it walks down the microtubule tract for anterograde exoplasmic transport. And then dynein is the protein molecule that moves the substances back from the terminal buttons to the soma. So important to keep those two straight. So if we just had neurons in our brain, they would be kind of a mishmash of connections, and we really need some sort of a support system or some sort of a cell to stabilize the neurons or provide structural support or scaffolding for the nerves. And so we have what are called neuroglia, which is actually, I think, Greek for nerve glue. And they provide physical support, control nutrient flow, and they're involved in phagocytosis and the immune system functioning in the brain. The neuroglia, or sometimes just glial cells, or glia, consist of three principal types of glial cells in the central nervous system, and that would be the astrocytes, the oligodendroglia, or oligodendrocytes, and the microglia. The astrocytes are sort of star-shaped as in nature, and they provide physical support. They wrap around a lot of neurons to kind of give them some support. They also remove debris. They're very metabolically active, and they also transport nutrients to neurons. They're actually a very important source of energy for the neurons, because they can actually extract glucose from the blood vessels, because they also wrap around blood vessels, and they can pass that glucose on to the neuron. But it can also, in times of low glucose, it can produce lactic acid or lactate, and then that will also give the cell a source of energy using the lactate. The oligodendroglia provide physical support, and they form a myelin sheath around the axons in the brain. And we have an example here of the oligodendroglial cell. You can see one oligodendrocyte is wrapping around many neurons to form a myelin sheath around the neuron. Now, interestingly, there's a corresponding type of cell in the peripheral nervous system to form the myelin sheath in the peripheral nervous system outside the brain and the spinal cord, and that's called the Schwann cell. And here we have one Schwann cell will wrap around one neuron. So it's a one-to-one connection for the Schwann cell, and we're talking about cell to neuron, and a many-to-one connection when we're talking about neurons to the oligodendroglial cells within the peripheral nervous system. So important to keep those two separate and know the difference between the oligodendroglia and the Schwann cell. The microglia are involved in phagocytosis and brain immune function. They're the brain's innate immune system, and they're really kind of the cleanup crew for various toxins that build up in the brain or injuries that occur in the brain, and we'll see in just a second how they do that. Now, important to know that the glial cells are unfortunately frequently associated with brain tumors, and so we have brain tumors that are called oligodendrogliomas if they result from an abnormal proliferation of the oligodendrocytes in the brain. That's a brain cancer type of brain tumor that results from the oligodendroglial cells. An astrocytoma is another form of brain tumor that involves the astrocytes. We also have microgliomas, and perhaps the most severe of all of them, Glioblastoma Multiforme, which is again arising from the glial cells. I think John McCain, the senator from Arizona who passed away in last year, had Glioblastoma Multiforme. Most brain tumors occur in adults between the ages of 50 and 60, with a median age of 57. It's delightful for me because I turned 57 this year, so hopefully I'm over the hump. But again, this is kind of, from a neuropsychologist's point of view, important to know how some of these different types of brain tumors are named, and that just tells you the glial cell that they arise from. So taking the astrocytes more in specifically, they provide structural support for neurons, as I mentioned, in the central nervous system. They're involved in neuronal metabolism, as I just mentioned. They use their connections with blood vessels to extract glucose, and they also are able to produce lactate as well to provide energy for neurons. They help participate in reuptake of neurotransmitters in addition to the neurons themselves. And very importantly, they contribute to the formation and the functional regulation and the repair of the blood-brain barrier. They're a very important constituent of the blood-brain barrier because, again, they help wrap around the blood vessels that are within the central nervous system. Sometimes, if there's injury that involves the astrocytes, they will produce what's called a glial scar or a hypertrophic cytoplasmic process. And this glial scar results from a traumatic injury or sometimes an infectious injury. And oftentimes these glial scars are the source of seizures in epilepsy. And the glial cells also regulate the chemical composition of the fluid that surrounds the glial cells themselves and the neuron, the extracellular fluid. And just sort of indicated that their close proximity to the blood vessels is important for extracting glucose from the blood, and they can either store it by converting that glucose to glycogen, and that glycogen can actually then be converted into lactic acid or for energy down the line. Or it can actually use lactate to provide energy for the neuron. You see this one astrocyte, again, star-shaped type of cell, and it's wrapping around the blood vessels, as you see here. The formation, it helps in the formation of the blood-brain barrier because in the normal capillaries in all of the body except for the brain, there are these little gaps in the capillary that actually allow substances in the blood to flow through. And so a toxin could potentially, outside of the brain, could potentially flow through these little gaps. In the brain, the capillaries have a different type of formation, and although there are gaps, the gaps are much, much smaller. And so it's harder for large molecular weight toxins to get through those little gaps in the capillaries in the brain. And because the astrocytes surround and kind of further seal up those capillaries, it helps keep those the substances from coming out of the capillaries in the brain to produce damage to the brain. Now, the blood-brain barrier is a semi-permeable barrier between the blood and the brain. There's a weakness in one area of the brain called the area postrema, and that's by nature's design. This weak area would allow toxins to seep through and stimulate the neurons in that area postrema, and that would stimulate a vomiting reflex for the organism to try to get rid of the contents or the toxins that they ingested. And then it regulates the chemicals that enter the brain from the blood. It only allows things like oxygen, I think glucose gets in. Only low molecular weight types of compounds will be able to pass through the blood-brain barrier, and helps maintain proper composition of the fluids inside and outside neurons. And this just kind of further explains the blood-brain barrier. Again, oxygen, carbon dioxide, amino acids, a few sugars like glucose, and many lipid-soluble substances like anesthetics, or what is able to cross the blood-brain barrier. And that blood-brain barrier is produced by those two factors: the very tight junctions between the endothelial cells lining the CNS capillaries. The endothelial cells also contain proteins and molecules that are not found in the capillaries outside the central nervous system. So there are actually receptors or channels, essentially, that will transport these things like glucose and amino acids across the blood-brain barrier into the brain for nourishment. And then the astrocytes, because they wrap around the blood vessels in the brain, those extensions coming from the astrocytes envelop the outer wall of the capillaries, further reinforcing them and making them less susceptible to leakage. That also helps the astrocyte maintain, regulate, and repair the blood-brain barrier if there's damage there. I mentioned that area postrema in the lower brain stem, and there's tight junctions between the cells of the capillary walls normally, but in this area it lacks the tight junctions, and it's more like capillaries outside the central nervous system. And the pineal gland and the hypothalamus also have no blood-brain barrier, so those are kind of the three main weak areas of the blood-brain barrier. Otherwise, the brain is remarkable in terms of protecting itself against toxic. Question?